So for today's midterm, oops, still too early in the term for that, sorry. But no, actually they're not graded either. That's another April Fool's joke. So I will, however, just for the point of having this as good practice, nice to have questions. These are going to be very similar questions to ones that show up on exams. So today's clicker question, which is actually not graded. The extracellular part of the virus life cycle is the virion capsid, nucleocapsid envelope, or genome. And as I often say, a lot of this is for me to go back and review stuff, or if it's stuff I think I didn't do a terribly good job of explaining before. My insurance policy. Anyone still trying to find clickers? He said they don't count till next week, so why do I need to bring them? Yeah, we're at 40 so far, and usually I think I've got 80 people signed up for the class, so I guess half of you still don't have clickers, or something, or didn't bring them with them. Case may be. Okay, it looks like we're not getting any more, so let's go ahead and stop. What do you think? Let's show results. Here we are. Yes, so extracellular part of the virus life cycle is the virion. And I will try to use virion when I'm talking about the extracellular part of the virus. A lot of people, me included, will say virus instead of virion when they really mean the virions, just the extracellular part. And the important thing here is really that viruses are not just the virion. Just the virion by itself, as you could think of this as being like a seed when you're talking about a tree, or an acorn for an oak tree. And that is a nice analogy that I like to use in terms of thinking about the process. No one would care if Ebola were just a virion. It's doing something to its host in that part of the virus life cycle. So that's really the important part here. Capsid is the protein which is involved in making that virion. And usually it's going to be protein. Nucleocapsid is just what's included very often inside enveloped viruses. And that's, again, going to be the outside of the virion, not so much the outside of the virus. And then the genome is what's left on the inside. So I can actually select the correct answer here, even though, for those of you who got here late, it's the 1st of April. So these are, in fact, not graded. And the quicker questions on Friday will also not be graded. And yeah, that's the 3rd of April, so it won't make a big difference. So the standard part of the virus definition, not used by Luria, is extracellular particle, parasite. Viruses have nucleic acid genomes, and A and B, or A, B, and C. Again, Stedman's favorite kind of exam questions. Although, A and B, not C, or all of the above except for P and Q. And again, since it's not graded, I can give you another hint. It's one of the things that Stedman doesn't like about normal virus definitions. And we could say most virus definitions here. I got one more. Got up to 41. $42. Well, 42 is the answer, right? To the life, the universe, and everything. So we may as well stop there. And parasite or extracellular particle and parasite. If you go back, and I didn't pull up his quote, but he talks about this particle which is made and that particle which is being moved. And so I understand that as being part of the extracellular part. Parasite is definitely something that he does not talk about there. And in my completely biased opinion, I think that most viruses are probably not parasitic. In terms of, again, thinking about that whole replication or life cycle of viruses, many times when they're inside a cell, that's not necessarily going to be something which is negative for the cell. And in many cases, it's actually positive for the cell. And a lot of times, it's going to depend on your particular conditions that happen to be going on there at that particular time. So now, just for good practice, everyone who leaves after clicker questions can leave now. And you've all been sitting on the sides if that's what you're going to be doing. So let me just quickly choose this one. And today, I'll also just want to briefly review above and beyond those clicker questions. And no, there won't be any more clicker questions. You can put them away. What we talked about last time, a lot of it, again, is kind of review because we had the clickers. What is a virus? It's not just the virion. It's really a whole life cycle. Why should we study viruses? Not just because they make their hosts sick in some cases. I'm much more interested in studying viruses because they're everywhere, in the air we breathe, et cetera. Yeah. Sure. The answer was B, yes. So it doesn't call it a parasite. Luria's definition of viruses does not include that it's a parasite. Extracellular particle was A. So A and B. They're also not just extracellular, right? So wouldn't it have been... But he also, if you keep reading down the bottom. Specialized particles that transfer the genome to other cells. So there has to be something extracellular particle which is moving it. That's why I give you these questions beforehand. And again, this one doesn't count. But the extracellular particle is part of his definition. It's, yeah. So that's the last part of it.
I guess I was saying that it wasn't just exclusively that.
Yeah, well, what's not part of it. Both. Right. Yeah, so it's not both of those. Right. Okay. So yeah, A and B, A or B. Again, these are, we can discuss these questions, but yeah, these are typical kinds of Stedman questions. So consider yourselves warned. I'm sorry, they're not the best questions by any stretch of the imagination. I probably made them up after having too much to drink late at night. It's that nasty secret that professors should never tell you when they put their exam questions together. The clicker questions are nowhere near as well proofread as exam questions. So at least hopefully that will help some of that. Okay. Other questions, comments, worries? Yeah. When is the midterm? Pardon? What's the frequency? AA. So it should be their standard frequency. Sorry, and thanks for reminding me about that. It's the frequency for these AA. And yeah, I will post clicker scores at the very latest at the midterm, but apparently there's some new software with D2L that we can upload them much faster. So hopefully by next week I'll also be able to make sure that you're getting all of those things registered. So again, questions about frequency and so on and so forth we should be able to take care of. There doesn't seem to be anyone else using these kinds of clickers around here right now, so it should work okay. Unlike down at CLSB, and I'm sure some of you have experienced some of that there as well. Okay, yeah, so what's a virus? Again, this whole life cycle. Why we should study the viruses? Because they're everywhere. Not necessarily because they're making us sick, although that's what we know most about those kinds of viruses, and the ones we'll focus on certainly for the last part of the course. Virus diversity. they're clearly ridiculously diverse, not only in terms of their structures, of the virions, but also in terms of the virus genomes. Most of virus genomes, and I would say that for really the majority of environmental viruses, don't match anything else that are present in the databases. And they're wonderful tools for understanding the host that they've infected because they use, and again, getting back to Luria's definition, intracellular machinery. So they're using host machinery to replicate. And so a lot of the differences we'll see between the different viruses is how much of that different host machinery is being used. In some cases, they use almost none of the host machinery. In other cases, they use almost all host machinery. And as we'll see later on, a lot of that has to do with how big the genome is. If you've got a really big genome, you don't need very much of the host machinery. If you've got a really small genome, you do depend a lot on the host machinery. Where they come from, actually, we really don't have a clue originally. Again, simulations, you get parasites that come up in these kinds of things, or I guess parasites in the case of looking at these simulations, but really kind of mutualistic environments, and I like to think of these as a symbiosis. And symbiosis is really a whole range of things between completely parasitic and completely mutualistic. And so if you think about a symbiosis, I think of viruses in much more of that kind of definition. And again, you know, why they're important to study, because they're totally cool. But again, I think mostly because of the ubiquity and diversity rather than the actual thing that most people study viruses about. So today we're mostly going to talk about how you can figure out how many of these, basically invisible to naked eye and light microscope, with a few really interesting exceptions, we'll talk about right at the end, of these particles. How do you study something which is basically too small to see? The main one there is going to be the plaque assay, and this is just an example of the plaque assay over here on the right-hand side. This is a bacterial virus plaque, but it turns out that this is something that's used in all kinds of animal viruses. Basically, this process is used for as many viruses as we can use them for, and it works extremely well. One of the things that's also really important conceptually, and these are all pretty much conceptual aspects here. The plaque assay is a real thing, and in fact, people in the recombinant DNA techniques lab are doing this, even though the stuff they're using is not recombinant at all. But the idea is a one-step growth curve, and anyone who's associated with my lab knows that we're trying really hard to do these. The idea of a one-step growth curve is basically to try and analyze a single virus, and again, I'm thinking about virus in the whole process, from virion through the infection cycle and making more virions. The problem is, at least with current technology, we can't study one single virus and one single host. And so you want to look at multiple viruses infecting multiple hosts all in the same time in the same replication process. And the idea there is then once you can do that, then you can look at what is called one-step growth curve, which is a bunch of viruses infecting a bunch of cells, and on average, they're all going to be the same when they go through that one round of their life cycle. In order to do that, you have to have a high MOI, multiplicity of infection, and requires you know something about statistics. How many of you had statistics? How many of you abhor statistics? Again, I don't. This is all the statistics you're going to need to know for this class. This is the Poisson distribution, and it's really quite straightforward. And we'll go through it in a lot more detail. And that one-step growth curve really leads to an understanding of the virus replication cycle. And we may or may not get to virus structures. That's why I brought my nicely defaded soccer ball here. Last time we talked about how do you get a maximal volume for a minimal surface area. That's exactly this. So the Brazilians have figured it out, and the viruses have figured it out as well. I try and put in some of these big picture slides. A couple of years ago, people asked, you know, what's the big picture? Why should I care? So I'll throw a few more of these up here. Again, this is sort of nice review. How many, one-step growth curve, and to some extent virus genome replication strategies and structures. So very general kind of question here. As again, I didn't bring the textbook with me, but we move further on through the course. Most of the rest of the chapters are going to be individual viruses. This is true now for all viruses that we're talking about here. What do you need to know? Plaque assay, PFUs, plaque-forming units. We'll talk about these again in much more detail as we move along. MOI, multiplicity of infection, the Poisson distribution. In terms of virus genomes and how we classify them, it's all about the messenger RNA. No, it's not all about the base, all about the base. It's all about the messenger RNA, at least as far as virus classification is concerned. And then for virus structure, it's really all about keeping it simple, stupid. As simple as possible, because that seems to be a really efficient way of forming virus structures. And curiously enough, even the second largest virions that are out there use these icosahedral symmetry structures of putting things together. So even things that have genomes larger than some bacteria are using still some of these very same kinds of concepts. So how were viruses discovered in the first place? Basically, because they were really small. And that was about it. This was at the same time that Pasteur and Koch were coming up with spontaneous generation, Koch's postulates. You have to be able to infect an organism with a particular bacterium or disease-causing agent, isolate that agent from that infected organism, and put it back in. And so that was the whole process that was going on here. And basically, around 1900, people were looking at a lot of these kinds of diseases. And what they found was, really strangely, basically going through Pasteur's process of saying, hey, if we make something that's completely sterile or put it through one of these sterile filters, then it doesn't grow anymore and it can't cause disease. Well, a couple of guys, one in Holland and one in Russia, took these filters that would block any kind of growth and saw that they didn't block disease, which was really bizarre. And in theory, they didn't know what each other was doing. In practice, it's kind of hard to tell, but certainly around 1900, there wasn't much communication between Holland and Russia. Soon thereafter, this really pretty prevalent livestock disease, foot and mouth disease, also you could put through these filters. And basically, what people called it at that point was just something that you could put through a filter that still caused disease. And eventually, that became a virus. So Contagium vividum fluvium, they thought it was a fluid, so not particulate, because it went through all these filters. Vividum, which is actually alive and contagious, so something caused disease and can go through these filters, and then became eventually an ultrafiltrable virus. I mentioned last time that we're now 100 years of bacteriophage as of this year, based on Twort studies, which were done in England, and then Darrell's studies, who also claimed to have discovered bacteriophage, were published actually 2 years later, and we'll hopefully have a second 100th birthday party in 2 years. Anyone want to go to Paris in 2 years? So should be a fun party. So the next question that came up, of course, because of this is, well, okay, they can't go through filters. Are they alive or are they not alive? And this is something which people have continued to discuss for many, many years and are continuing to discuss today. If people ask me about are viruses alive, I completely obfuscate and say that viruses are part of life. And that still doesn't answer the question. But there was a lot of discussion, particularly at that time, and again continuing to today, about whether viruses are really alive. You can crystallize virions. Okay, virions can be crystallized. You can't crystallize life. Okay, well, but we hopefully now know that virions are only part of that virus cycle. One of, my major funding sources is NASA. They're very interested in discovering the origins of life on this planet and maybe life on other planets. They're also very interested in trying to figure out, well, if it's not a little green man on Mars, what is life? And how do you define it? And so part of that process has been with something which undergoes evolution. Well, clearly viruses undergo evolution. They're not going to undergo evolution in the virion, but through that replication process, they're clearly undergoing evolution and have genetic material that is passed along. So clearly, I would say that's much more of a definition for being alive. On the other hand, Eckhard Wimmer recently synthesized a poliovirus genome and then put it into a cell, was able to get it to function, and he has this wonderful chemical formula for polio. C332,652, H492,388, etc. No, I don't expect you to remember those numbers. But this, he basically threw that out there to say, well, if you can put a chemical formula on it, is it really life? Is it alive? Is it not? And there's a quite long discussion about this. If you think about the virus, okay, let's call them a replication cycle, not to call them alive, they're clearly undergoing reproduction as well based on a genetic system. So there are, back in the day, and you may even see, I may have some of these back on D2L, I used to ask a question on the problem set, which we don't have anymore because you didn't want one. Are viruses alive? And I took both answers, yes and no. And both of them were reasonable as long as a decent description. Yeah?
You said the virions can be crystallized. What about the other stages?
So generally, so the question is, virions can be crystallized. What about other stages in the process? In the actual production of virion stage, so again, this is really virions, which is still intracellular, some of those are quasi-crystalline. But again, it's the virion at that point when you're talking about it. But you could also crystallize small pieces of DNA. So, you know, what does that mean? But that was a lot of the discussion, particularly earlier on, is are viruses alive or are viruses not alive? And the person who crystallized tobacco mosaic virus, which was the first one that was found by going through these filters, was adamant that, oh, we can crystallize it, then it can't be alive. You can't crystallize something which is alive. So this is, again, is still, I think, a very open and interesting question, and again, probably discussed with considerable consumption of alcohol. Some really cool things you can do with viruses that a lot of people don't necessarily think about. Phage therapy, anybody heard about phage therapy? Basically, the idea here is to use bacterial viruses to treat bacterial diseases. And hopefully probably all heard about antibiotic-resistant bacteria. MRSA, for instance, is a really nasty problem. There are a number of viruses which specifically will infect and kill MRSA. And so the idea here is to use the virus to kill the bacteria that's making us sick. And there's a group up at Evergreen State College, just up the road in Washington, who's done some really, really nice work on this. There's also been some great work that was done in the former Soviet Republic of Georgia, now the Republic of Georgia, where they've been looking at using bacteriophage therapy. It's becoming a pretty big deal. There are a number of companies now as well. And that link I gave you to the phage birthday party, there are a number of companies who are doing this now, trying to commercialize these bacteriophage particularly for antibiotic-resistant bacteria. Gene therapy, probably 80 to 90% of gene therapy trials use viruses in that process. And as we mentioned last time, you know, my favorite definition of a virus is a bag of nucleic acid. Well, that's what that bag does, is move genes around. And so gene therapy very much is involved in reprogramming those viruses in order to move the genes that you want as opposed to the virus genes, changing the nucleic acid which is present on the inside. Nanotechnology, I like to think of virions as nature's nanoparticles. They're generally much better designed, again, from my totally biased point of view, but they've had clearly millions, probably billions of years of evolution to get to a certain number of solutions to making nanoparticles. And a lot of people are either using virions directly for nanotechnology or using this as, you know, inspiring the kind of design that they're making there. As I mentioned before, HBO program just a couple of nights ago, cancer therapy. They're using engineered polioviruses to now treat, I'm trying to remember which cancers they were. I think this was leukemia that they were using for that. But there are a number of different processes here. And the return of the phage is just a posting that I put on D2L. I'm not going to be testing on that per se, but if you're interested in some of the things that are going on there, that's a good short review. And then if you're interested in more, again, take a look at the talks at the phage birthday party. So not just cool things you can do with viruses, using viruses as tools. Basically, everything we know about molecular biology comes from the study of bacteriophage. DNA is the genetic material, recombination, promoters, replication, et cetera, et cetera, et cetera, et cetera, et cetera. Almost all of that comes from the study of various different bacteriophage, and in fact, a lot of that, in fact, just from studying bacteriophage T4. So that has been really pretty revolutionary in terms of what we know about molecular biology. And one of the reasons there is, again, getting back to what these viruses are doing. They're using the cellular machinery to make more virus. Well, it's usually completely reprogramming the cellular machinery, and we'll see how that works as we move through some of the other viruses through the rest of the course. But it's great because they're relatively simple, and you can understand what's going on with the virus, and then it turns out, apply that back to the host cell. Eukaryotic molecular biology, a lot of tumor viruses were used for this. Turns out most cancer is, in fact, not caused by viral infection, but a few cancers are. Papillomavirus is probably the best example of those. But here, again, understanding the process of how cancer works, oncogenes discovered through the study of tumor viruses. In terms of looking at replication, a lot of the textbooks even still have, at the origin of replication, they have large T right next to the origin, the replicative helicase. Well, that's not the cellular replicative helicase. That's the replicative helicase for the virus SV40. And that's how these replication origins were originally found. Retroviruses, again, anywhere between 8 to 40% of our genome is based on retroviruses and their replication. So these are just, again, some of the breakthroughs, some of the things, just a very small tip of the iceberg kind of thing about things you can do with viruses and what we've learned from studying of viruses. So this is great, it's wonderful, but how do you actually know how many of these things that you can't see you have around? So that leads to these different processes for studying amount of viruses, basically gone from the hardest and most expensive to the easiest. Animal work, which is, okay, in the most extreme case, bipedal upright animals. But basically looking at how viruses spread in animals in different populations, et cetera. That gives you an idea of numbers of viruses. It's really important if you're actually trying to study pathogenesis. Any kind of plaque assay, something like this we'll talk about before, tells you absolutely nothing about pathogenesis. So if you care about what kind of diseases are being caused by a particular number of viruses, you really have to do animal models. They're expensive, they're messy, they smell, but if you care about pathogenesis, you really have to go down this mechanism. Electron microscopy, I mentioned before, most virions you can't see in the electron microscope, with now, in the last 10 years or so, a couple of exceptions. First time I ever saw viruses, or at least pictures of viruses you could see in the light microscope, I was just blown away. This was really cool. I could actually see the virions themselves. But it's only 2 or 3 now that you can do that with. For the most part, you have to have an electron microscope. And electron microscopes were only developed in the late 1930s. So actually seeing virions, we've only been able to see in the last 60, 70, I say back calculate 80 years now. God, it's already 2015? No way. Okay, so, but since the development of the electron microscope. Anyone who's done electron microscopy knows that this is also incredibly difficult. You see a lot of junk. And as we get to a little bit later on, It just is going to show you the virions. You have no idea if that virion is active, if it's functional, etc. You're just going to see a particle. Yeah?
What kind of concentration of virions do you have to have in the electron microscope in order to like see them without seeing too many? I mean, like you want to have enough on whatever your plate is or whatever that you can like actually find it, but like not so many that it's crowded and you can't identify it.
Yeah, so the question, sorry, I'm just going to repeat this because it's being recorded. Now, how many virions you need to be able to see in the electron microscope? And the answer to that is basically it depends on how lucky you are. But usually you need a lot because what you're looking at in terms of an electron microscopy, transmission electron microscopy, which is basically the process that you need to use, you use these grids which are about 3 millimeters across, and you put a couple of microliters on that. And then on each of those grids, they're separated into, depending on the grid, about 400 little squares. And so you're looking in one of those little squares at a magnification of usually 20,000x. And so you have to have a pretty high concentration of particles to be able to see them. Usually on the order of, and we do this in my lab, 10 to the 8th, 10 to the 9th per milliliter to be able to actually see them. So 100 million to a billion per milliliter. So this example, for instance, over here, this 10 to the 11th one, you can see those particles really quite well. We've also gone to even higher concentrations, and actually I should have brought that picture. There's one posted outside my lab of just sort of wall-to-wall virus particles. But you can still identify them as virus particles. That particular one was probably a concentration of about 10 to the 13 per mil. So you need a lot. Basically, is the answer to that. So what people do, mostly because, again, animal work is really expensive, hard to do, electron microscopy, you need a whole bunch, and you don't know if these are actually functional or not. Most people, if you can, are going to do a plaque assay. And so plaque assay is basically shown up here in the corner in a cartoon version. You take your virion, this should be suspension, and dilute it a whole bunch. And we'll see why that is in just a second. And you've got really very, very large numbers here. And then eventually take those dilutions, mix them together with a host, and then let everything grow, and then see where you have virus infection that's taken place, each of these plaques. This incubation period is really important because what you're doing is you're allowing multiple life cycles of virus infection to take place. And so the idea is here you'll have one infectious virion interacting with one cell, then that will undergo its full life cycle, make more virions, which will infect the cells right next to that first one, which will then make more virions, infect the cells next to that, et cetera. And eventually, you'll end up with a visible area of 6 cells. And in the case of a lot of bacteriophages, you've literally killed off the cells in the process. And as I mentioned before, very often you've got these very high amounts of infectious virus particles here, or virions, 10 to the 11th. So you have to do very large amounts of dilutions here. And then what you do is just count the number of these what are called plaques, and then back calculate through the number of dilutions to give you an idea of the numbers that you started with in the first place. Another way of looking at this, here's your virus dilution. You mix it together with your host cells. You put it in top agar, and the important thing here with the top agar, particularly for bacteria, something like E. coli. E. coli loves to swim around, right? So if you're going to have an infection of one cell that's going to infect the cells next to it, which infect the cells next to it, then if these cells are running away, you're not going to have a nice plaque. And so what the agar is doing is stopping those cells from swimming away and not being in the same place. And again, this incubation process here is really critical because it's multiple rounds of virus replication that you're looking at. Any one of these plaques here, and these are in fact plaques from lambda bacteriophage, contain probably 1 million virions, 1 million to 10 million virions. It depends on the actual plaque. So these are the really pretty plaques that you see in textbooks. These are real plaques that I actually made in the lab from that Phi-X 174, that tube that I showed you that had 10 to the 11th virus particles, infectious virus particles in it, virions. So here, counting individual plaques, each of the dots here was where I counted one. Here it looks like one I ended up missing. Got 67 on this plate, 1 dilution, 3 on this plate. This was supposed to be 10 times less than that. Gives you a bit of an idea of the variability that you very often get in these. Same thing down here. This was actually a different virus prep. 81 plaques here and 19 there. So you basically will average across your different dilutions. You'll do multiple different plates, and that'll give you an estimation of the number of infectious particles that you have in there. Yeah?
So your plaques are basically just holes in your lawn then?
Exactly. And so what this is it's some indication that you had a virus infection at one spot at one time, which is now replicated and infected all the cells around it. So you assume that each of these holes, as often call them, in a lawn is due to what was one infectious virus particle. One of the things which I forgot to mention earlier, so phage or bacteriophage. Why are they called bacteriophage? So what's phagos? Any Greek background? To eat. So the eaters of bacteria. That's what these bacteriophage are. They eat the bacteria. So they've eaten a hole in your lawn of bacteria here. So gone through this works great for bacteria. It works quite well for mammalian cells that you can grow as monolayers. Anyone who's worked with mammalian cells know that there are lots of cells you can't grow very well as monolayers. And there are lots of viruses that there aren't good plaque assays for. I thought that these plaques were really ugly until I tried to do some plaques on some mammalian fibroblast cells, and they're way uglier than these and a lot harder to count. But still, much easier than doing, okay, I put a certain dose into a certain animal. It had a symptom, but 10 times less in a different animal had a different symptom. Put 10 times less in this animal, didn't have any symptoms. How do you get any kind of statistics out of that without going through a whole bunch of animals? So that's one way of doing it. Another possibility, if you don't have a good plaque assay, is something called hemagglutination. We'll talk about the envelope proteins of particular viruses a lot more later on in the course. Last time we talked about the envelope proteins. These are the viral proteins that are sticking in the membrane on the outside of the envelope. Many of these bind to cell surface, what are called receptors, often present on red blood cells. And so the idea here is that if you have a virus dilution right here, mix it together with red blood cells, if there's something on the surface of the virus that will bind to those red blood cells, what it'll do is end up cross-linking those red blood cells. And you end up with these cross-linked red blood cells here in the presence of virus. In the absence of virus, if you have the right kind of plate, which I didn't the first time I did it, they'll end up right at the bottom of your tube. And so the idea is basically shown down here at the bottom. Here is a example, let's see, in this top row is a positive control. You can see that these guys look pretty aggregated. The second row here, B, is a negative control, no virus added whatsoever. So you have all these little spots at the bottom. And then C through H are all different samples that you're testing to see basically, is there virus in there or is there not virus in there? The way that you get an idea how much you have here is just doing dilutions. And the serial dilutions basically take half of what's in here, add it to this one, half of that one to here. You just end up with twofold dilutions, 1 to 4, 1 to 8, 1 to 16, 1 to 32. et cetera, across here. So let's take a look, for instance, let's take one of these here, F. At very low dilutions, you seem to have something there, but not very much. D and E are nice examples of actually quite a lot of virus. You're seeing good cross-linking up to about 512, almost to 1024, and then eventually you're not seeing any cross-linking out at 1 to 10,000. One thing you'll notice here is that these numbers are way, way, way lower than in those plaque assays. You know, plaque assays, we're talking 10 to the 8th, 10 to the 9, etc. Those are hundreds of millions of billions. Here's our tens of thousands. But that's because you need multiple virions to cross-link these different blood cells to each other. So it's, in fact, a not very sensitive technique for doing this process, but it has a couple of really major advantages. It's very fast, it's cheap, it's pretty easy to modify. You can put in antibodies that'll block kind of interactions with various different viruses, etc. And the main thing is it's really easy, and it's so easy even I can do it. So here's an example of a hemagglutination assay, which I did a couple of weeks ago in the lab, looking at influenza virus and influenza virus with red blood cells. This is how it started. These are just mixed red blood cells with various different amounts of the virus. This is how everything starts. You leave it for a couple hours, and in my negative control, you see all of these spots down here at the bottom of the tube. So these are the ones that didn't have any cross-linking going on. Here is my positive control, where I dilute the virus out to about 1 to 64 before you start to see this spots down at the bottom. And then these were a couple of different treatments that I did, silica coating of the viruses, and you can see that they're slightly different than our positive control here. So yes, it's a possibility. It's not the prettiest, but if I can do it, it's got to be relatively straightforward. So that brings us to another issue, and this was actually getting back to discussions we had about electron microscopy, and that is that the number of plaque-forming units or hemagglutination-forming units, whatever you want to call them, is often a lot less than the number of virions that you can see in the electron microscope, that you can count some other way. Way back last lecture, when we looked at those fluorescent spots, the really tiny spots that you see in seawater, you see in soil, you see in any environment, each little spot says, hey, there's nucleic acid here, and it's in a small place. But that doesn't tell you whether it's infectious. And so you have to have some kind of infectivity assay, like a plaque-forming unit, to say that that little dot, that little particle which you're able to find, is in fact infectious. And a lot of the virus detection techniques that are out there, even for diagnostic purposes, don't actually count the number of infectious viruses, those that can go through that whole process. They're often just counting numbers of genomes. They're counting numbers of particles. And so I think you have to be very careful about thinking about exactly what you have and what's there. It's a lot easier to count particles. It's a lot easier to count genomes, particularly with quantitative PCR, but that doesn't tell you if you actually have infectious particles. And why is that? Often, and we'll see this a little bit later on, even when we look in the electron microscope, not all particles are intact, and the intact nature of a particle, as far as the virion is concerned, is going to be how it's going to be able to interact with the outside of the cell. How is it going to be able to infect? How is it going to be able to get in to the cell? If it's not completely properly formed, it's not going to be functional. It's not going to be infectious. Often you'll have a defective genome. We'll talk about virus mutations, particularly for RNA viruses, have very high mutation rates. A lot of these are going to be genetic changes. Many of these genetic changes will lead to a non-functional genome. Well, if you're just counting genomes with your qPCR assay for HIV, it's like, oh, look, there's HIV there. Well, yeah, there's a genome that looks like HIV there, but is it really functional? You don't necessarily know. If you're just using EM, empty capsids, the packaging process, putting the genome into the bag, the nucleic acid into the bag, making your virus, is not terribly efficient in some cases. It turns out bacteriophage is extremely efficient, most eukaryotic viruses, it's not very efficient at all. If you take a look at HIV, just to take one particular example, it's anywhere between 1 in 100 or 1 in 1,000 virions is actually infectious. So it's really very, very low. And then, if you're thinking about infection processes, depends on sometimes your antiviral defense. You're doing an animal study. If you're infecting an immune-compromised mouse, versus a non-immune compromised mouse, you're going to get a very different number in terms of how many viruses you actually happen to have there. So this is basically a caveat to bear in mind. And again, some of the most extreme, you can have only 1 in 1,000 of your virions actually be an infectious virus or part of an infectious virus cycle. Why is this so important? Well, if you want to study virus infection, of course, you want to have infectious viruses. In order to look at infectious viruses, you really need to look at a whole bunch of infected cells at the same time, lots of viruses that are going through their whole replication process simultaneously. And that brings us to the whole concept of the one-step growth curve. And basically, all that this is a synchronized infection where you have, here are virus, infectious, multiple particles. You infect the cells. You try and get all of the cells infected as close to the same time as you can. And then you just wait for that replication process to go through, starting out with, and now we're just looking at plaque assays here, start out with certain number of plaque-forming units per host cell. Usually you want to use 5 to 10, so relatively high number of PFUs per cell, which is just the multiplicity of infection, number of PSUs per host cell. That then goes down to basically 0. We'll talk about why that's not exactly 0 a little bit later on. And then, just following this black line, after a certain period of time, in this case 40 hours, but it just varies from virus to virus. In the case of bacteriophage T4, it's about 30 minutes. Here for this virus is about almost 2 days. Now you see maximum virus production right here. So this is your one-step growth curve. Doing your infection over time, waiting for the virus production, or say virion production, PFU production to be the highest. Why is this such a useful thing to look at? Well, because basically you can now, over this time period, look at this infection. So all the cells were infected here. They start to make more virus genomes, more virus proteins. This particular case is looking at a T antigen, eventually making more of the viral nucleic acid, and then finally these infectious virus particles. What we're looking at here in the black line is literally just a plaque assay over time. Start out with, again, about 10, 5 to 10, and in this case you end up with about 400 or 500 per cell. So what that tells you is that each of the cells has produced about 500 viruses. We'll talk about a few more things that you can get from these processes. This is just another way of looking at this. Again, you add your virus to your host cells. Very often you do this with a concentrated host cells because you want to have as small a volume as you can start with because you let everything grow. Now, the viruses are growing, the host cells are growing simultaneously here. Once you add a whole bunch of virus to the cells, you don't want any extra virus left over, because anytime you have a cell that's growing, it's becoming reinfected, you've got multiple viruses infected at the same time, you want this to all be simultaneous. Infect it once, and then follow the whole process. And again, follow the plaque assay over time. And this is just another way of looking at that. Here you add virus. Here you end up with virus at the end. Most of the interesting stuff that we're going to spend almost all the course talking about is all of what's going on here. It's making more of these virus particles. starting out with what's called the early enzymes, making the nucleic acid, making the bag that they're going to go into, and then putting that bag together. This is also called the eclipse phase of infection. We'll talk a lot about the eclipse phase, but that's because you don't see any infectious virus particles during this process. So here, until you start to see this rise in the number of PFUs, here we have no infectious particles which are actually there. Look at some real examples now of some one-step growth curves. These are various different viruses. Here's adenovirus at the top, Western equine encephalitis in the middle, and then just bacterial growth for comparison down here. All of the y-axes here are log scales, and just time across here on the x-axis. Bacteria, okay, 1 to 2 to 4 to 8 to 16, etc. Always undergo binary fission. One thing to notice here is that all of these curves are much faster than binary fission. So that tells you already that infectious virion production is very different than binary fission. We already talked about this eclipse phase. This is before you have any infectious virus particles which are being produced. There's something else which you see in particularly viruses that don't have envelopes on the outside. It's what's called the latent phase. And sometimes the latent phase actually has a different time compared to the eclipse phase. And that you can see if you do an experiment, which I actually mentioned here on the last slide, is if you artificially break open the cells. If you artificially break open the cells and then do a plaque assay, what you can see in some cases is that the virions that are still inside the cell are actually infectious, but they haven't gotten outside of the cell yet. And so there's this difference in time between the production of an infectious particle and those infectious particles actually getting released. That almost always happens with naked viruses, those that don't have envelopes around the outside. And the reason for that is just the numbers build up of these protein and protein-only capsids inside the cell. They somehow got to get out. Well, how do they get out? They usually get out by bursting open the cell or producing some kind of protein which makes a hole in the outside of the cell, and those then get released. On the other hand, enveloped viruses, they've got a membrane around the outside. What's on the outside of all cells? Membranes. And so that process, and usually these viruses are picking up something which is present in the plasma membrane. Those are then being released through the plasma membrane, and you now only have virus particles once they've gone through the plasma membrane. What that means is if you take an infected cell and break it open, there are no infectious particles in the inside because they haven't had the chance to pick up that membrane and actually leave the cell. So if you see something like this, where you've got intracellular and extracellular identical to each other in a one-step growth curve, almost definitely going to be an enveloped virus particle. If you see a difference, then it's usually going to be a naked virus particle. Yeah?
How does that fit in with the plaque assay if you're lysing these cells? Because if you're lysing them, then how do that make a plaque? I guess I'm
Okay, so the key here, that's a good question, and there seems to be some confusion about this as well. So the plaque assay is you take a sample from your one-step growth curve, and then you put it in a plaque assay. So it's a completely separate thing that you're doing there. So the plaque assay is separate. The plaque assay is just a way of getting numbers. And you could even also just do a qPCR assay, or you could do an electron microscope assay to just get the numbers. And so it's just a way of counting actual numbers there. Usually, again, you're going to use PFUs, but they're completely separate. Yeah.
So PFU can come from different numbers, not just from a plaque assay counting?
No, so a PFU is literally a plaque. So, but you get virus numbers some other way. But it's just a numbers. So anything that's going to be on your y-axis here is just numbers. Yeah?
It's like a liquid culture. You're growing bacteria in an environment at 5 minutes and 1 hour.
5 hours.
Right.
Run it out.
You take some of it out and you do a plaque assay on it. Yep, exactly. Okay, there's a couple of other things that you can get from these one-step growth curves. One thing that a lot of people will talk about in terms of viruses is what's called the burst size. And all that is the number of plaque-forming units you have at the end of your one-step growth curve divided by the number of plaque-forming units that you have during the latent phase of your one-step growth curve. And that will give you the number of virions produced by one infected cell. And that can vary anywhere from 1 to 2 to hundreds to thousands that any of these individual ones are being put on here. And so this, by the way, is figure 213 from the Flint textbook, which should be available in the library if you want to go and take a look at it there. So that brings us to everyone's favorite statistics, talking about infection processes. If you want to do a one-step growth curve and get all this information from it and study what's going on in terms of virus replication, you need to know that you have a synchronous infection. And so that brings us to the Poisson distribution, which is a statistical description of random interaction of different particles. And so you have a virion, you have a cell, and that interaction is very well described by this Poisson distribution. And so what we're looking at here, and we'll look at the actual equation in just a second here, but we're looking at the fraction of P, which would be one set of particles, that have been infected by or interacted with the number of, we're looking at virus particles here, actually interacting with each other. So that's the actual number they're interacting with each other. M is the number you start with. So the number of PFUs you put together with number of cells, also known as the multiplicity of infection. So we've talked about this, hopefully, almost ad nauseum now. Number of PFUs per number of cells is your MOI. Now, why is this important? And this is just a graphical description of what we're talking about from a Poisson distribution. So you have an MOI of 1. You've got 5 nice little blue virions and 5 poor unsuspecting E. coli's. Sometimes you're going to have, with 5 and 5, you'll just have what you expect. One virus particle interacting with one cell. But sometimes you'll have 2, and sometimes you'll have none. This is just, again, your statistical distribution. If you start out with an MOI of 2, so now you've got 10 of these virus particles and only 5 E. coli's, you'll have some that have 1, some that have 3, some that have 2, etc. If you have an MOI of 5, then almost all of them are going to have some, but not very many are actually going to have 5. You'll have 4, you'll have 6, etc. And the way you can look at this now is by looking actually at a Poisson distribution. And so this is P of A, I usually use this P of K, here at a M of 2, or an MOI of 2. And what you can see is the number of cells here that have 0 particles associated with them is about 13%, even though you've got twice as many virus particles as you have cells. About 27, 28% have 1, 27, 28% have 2, etc. And then that goes down, more numbers you get. Now if you go to 10 times as many virus particles as you have cells, about 12% are going to have 10, but a very, very small percentage are going to have 0 here. You can do the calculation. Here's the Poisson distribution here. P of k is equal to e to the m, m to the k over k factorial. If I give you an equation like this on an exam, I don't expect you to remember this. I'll actually give you these numbers. Don't worry about it. But what's really important here is how many of your cells have 0 viruses that are associated with them. Because those are going to be uninfected. And if you're trying to do a one-step growth curve where you have simultaneous infection of everybody at the same time, you want to have this to be as low as possible. So if you look at an MOI of 1, so 1 virus particle per 1 cell, your p of 0 is going to be e to the minus 1, because that's, m of 0, 0 factorial, which is 1. 36.7% of your cells are not infected if you just have 1 virus particle per 1 cell. That's not going to be a synchronous infection by any stretch of the imagination. MOI of 2, 13%. Just showed you on that last slide. MOI of 5, 0.7%. That's pretty good, but it's still, not as good as you would like. MOI of 10, on the other hand, is 99.95 now are going to be infected. And so generally, you don't really care that much about what's uninfected. What you really care about is what's infected. So anything that's uninfected, 1 minus everything that's uninfected is everything that is infected. And so these are the numbers that we really care about. So 1 minus p of 0 are going to be everything that's got more than 1 PFU that's associated with it. So again, MOI of 1, 63%, 2, 86, MOI of 5, we're looking pretty good. Over 99% infected, MOI of 10, 99.995. Those people who are unfortunate enough to work in my lab know that getting enough virus to do this with is a real pain, right, Tian? So why don't we stop there, and we'll start with this virus replication process, because it's kind of a different concept on Friday.